Hey everyone, welcome back. So now we're going to talk about mitosis. So I've mentioned before, but let's remind you of what mitosis is. Mitosis, definition-wise, is the division of the nucleus. And when the nucleus divides, the chromosomes will also divide. And so the function of mitosis is to produce two cells that have identical DNA. So we start with one cell that is diploid, so 2N, diploid. And we produce two daughter cells that are also diploid, and they are identical. So we do this through mitosis and through cytokinesis. And cytokinesis is division of the cytoplasm. Okay, so let's look at the different stages of mitosis. The first stage of mitosis is prophase. And prophase is often separated into 2 or 3 parts. We have early prophase, prophase, and then prometaphase or late prophase. I typically just say prophase. So this is all this first step. And during prophase, the nuclear membrane, so the nucleus membrane, starts to dissolve. So we have this nuclear membrane dissolving or breaking down. The DNA that has already replicated starts to condense, forming those chromosomes. And so uncondensed, it's known as chromatin. So the uncondensed DNA, or the chromatin, condenses into chromosomes. So now we have these chromosomes that are held together at a region known as the centromere. The spindle fibers in the centrioles start moving. So centrioles move to opposite poles of the cell, and spindle fibers within those centrioles start to form. Centrioles are found in structures known as centrosomes. Centrioles move to opposite poles and spindle fibers form. So the spindle fibers then stretch out from the centrioles to the centromeres on the chromosomes. So let's look at this in your figure. So here we have in early prophase, the chromatin is condensing into chromosomes. The nuclear envelope is breaking down. The centrioles are moving to the opposite poles, and spindle fibers are coming out, okay? So it's early now in prophase. They're still moving to the opposite poles. Spindle fibers are moving closer and closer to the centromeres of the chromosomes. Nuclear envelope is almost completely broken down. Late prophase, spindle fibers have reached out and they're attached to all of the chromosomes at the centromere positioning. The centrioles are at the opposite poles, and the nuclear envelope is completely dissolved. So everything is ready for the next phase. So what is the next phase, you may ask? Metaphase is the next phase. So in metaphase, the chromosomes line up one on top of another. They line up at a region known as the metaphyseal plate. And so the metaphyseal plate, or the metaphase plate, is just an imaginary line in the middle of the cell. And the chromosomes line up there. They're puppeted there by those spindle fibers. So the spindle fibers are basically moving those chromosomes where they need to go so that they then can be prepared to be separated. So the next phase is anaphase. So sister chromatids of each chromosome are pulled apart. And they're pulled to the opposite poles. So the purpose of this, if you have, and I'm just going to draw it up here. We'll do the blue again. You have these two chromosomes, right? One from mom, one from dad. They're consisting of each of two sister chromatids. In anaphase, we're going to pull, so this is, I'm going to move this down here, okay? In anaphase, we're going to pull the chromosomes to the opposite poles. So that now we have one chromosome from mom and one chromosome from dad on each side. And then telophase can occur. So in telophase, I really do like to have a different color. Okay. In telophase, nuclear envelope reforms. So it reforms around the chromosomes. And as the nuclear envelope reforms around the chromosomes uncondense. They relax into their chromatin form. Sorry. During telophase, cytokinesis occurs. Actually, cytokinesis starts, and it started in anaphase, and it finishes in telophase. So we start cytokinesis during anaphase while the chromosomes are being separated, and then we finish cytokinesis in telophase. And so here we have metaphase, where those spindle fibers move the chromosomes to this imaginary line right here, known as the metaphyseal plate. Okay? So once that happens, now the spindle fibers pull the sister chromatids of each chromosome apart. That's an anaphase. And if you notice right here, there is an invagination occurring of the plasma membrane. That's cytokinesis. So cytokinesis is starting already. And then in telophase, new nuclear envelope reforms and the chromosomes are going to uncondense. Cytokinesis is going to finish. And we're going to end up with 2 daughter cells from 1 initial parent cell. And they're going to be identical. So if we look at mitosis in plant cells, things occur a little bit differently, but I actually am going to do that in my next video. So I will talk to you in a little bit. Bye.